خدا من فارسی یه صحبت میکنم بعد میرم به صورت انگلیسی. خوش آمد میگم به همه و مخصوصا تیم دانشگاه واشنگتن. سمپوزیوم خربارگری اثرات داروها بر روی بافت مهندسی شده که با حضور دوستان و همکاران عزیزم از دانشگاه واشنگتن سن لوئیس میسوری انجام میشه. من به خاطر اینکه این عزیزان کلا هیچ کدومشون در ایران اقامت ندارن، با اینکه دوتاشون ایرانی هستن و در واقع الان در دانشگاه واشنگتن در حال فعالیت هستن، یک خلاصه مختصری رو در ارتباط با کنگره میگم به زبان انگلیسی. بعد تعریف میکنم آقای دکتر گایژنین رو و به ترتیب هر کدوم از عزیزان اومدن یه مختصر بیوگرافی در موردشون میگم و let me start the first symposium of preclinical drug development engineered tissue. Dear Congress, distinguished guests, and honorable participants. It's really an honor to have lectured together with world-known scientists participating and lecturing in the present symposium. Saying this, I salute and welcome you and all warmly congratulate Professor Syed Mehdi Rezayat, Chairman and Faculty Member of the School of Medicine, for his initiative and hard work to organize Congress, and Dr. Muhammad Qazi Khansari, Executive Manager of the Congress, for giving us the opportunity to successfully participate online today. From myself and our team from Washington, Missouri of St. Louis, I feel confident that you and will enjoy both the scientific program, and I wish you all the best and productive symposium. The 25th Iranian and 4th International Congress of Physiology, Pharmacology 2021 was held at the Tehran University of Medical Sciences, Tehran, Iran. This Congress is held every 2 years and is an event in which a basic science researcher in various fields of the physiology and pharmacology present and discuss the latest findings and innovation in various fields of physiology and pharmacology. Our symposium on clinical drug development, engineered tissue, focus on the latest development of the effects of drugs on various engineered tissue. In fact, the latest innovation and achievement in engineered tissue and the determination effect of drug present by my colleagues from Washington University of St. Louis, great researcher and scientists from famous university in the world, and of course from the Iran. Our first keynote speaker, distinguished Professor Gaijenin, Harold and Kathleen Faut, Professor of the Mechanical Engineering, lecture on the mechanobiology of tendon to bone attachment and repair. Let me at first introduce the Professor Gaijenin. Professor Gaijenin graduated BA Bachelor and MS from the Case Western Reserve University and MSc and PhD from Harvard University. His expertise is the study interface adhesion in physiology and nature, and he focused on mechanobiology, biometrics, quantitative image analysis, interface, and adhesion. Gaijenin is the faculty member of the Department of Mechanical Engineering, Material Science, and Biomedical Engineering in the McKelvey School of Engineering and Department of Neurosurgery in the School of Medicine. And he is the fellow of the American Society of the Mechanical Engineering, ASME, and American Institute for the Medical and Biological Engineering, and several other positions. Gaijenin studies interface and adhesion in nature, physiology, engineering. His current research focuses on interfaces between the tissue at the attachment of the tendon to bone, between cell in the cardiac fibrosis, and between protein structure at the periphery of the plant and animal cells. After introducing Professor Guy Jenin, I welcome to the Guy Jenin, and Guy Jenin, you can start your lecture. Move on, please, Guy Jenin.
Well, thank you so much. It is a great honor to be at this symposium. Vahid, it's great that you've been a longtime friend of Washington University, and it's an international figure in the research in this area. It's truly an honor to have the opportunity to present some research and get your feedback on it, and also learn from everybody here at this conference. Thanks so much. So I'm going to share my screen now, and I'm going and this often works, and I am going to then share some slides. Before I begin, I need to go through a couple of quick disclosures. I have to note that I do have financial conflicts of interest. I have a company called Cayley Vascular. I also have what are called foreign influence conflicts of interest in the United States, including having good friends in Iran. So great to be with my very good friends in Iran. So the topic that I'm going to speak about today is different from what was on the different from the abstract that I sent in, because Vahid very wisely said, perhaps you should speak about something relevant to the conference. So you'll hear from many of my very close collaborators and dear friends today. We're very interested in how fibroblast cells interact with cardiomyocytes and other cells to cause significant disease. And you'll hear from three world experts following me on this. You'll hear from Farid Ali Safai, you'll hear from Delaram Shakiba, and you'll hear from Elliot Elson. They're going to focus more on the mechanical aspects of the work that we all do together. I would like to focus instead on electrical aspects of what goes wrong in cardiac fibrotic myopathy. And we want to focus specifically on the issues that are druggable. So ask a question in terms of what goes wrong when cardiomyocytes interact with fibroblasts. What can we drug, and what can we learn from these engineered tissue systems? So forgive me for changing the topic, but it's now something relevant to the symposium. Apologies for sending it an abstract, totally unrelated. Before I get in though, a little bit of philosophy. So in the United States, and I suspect in Iran as well, when you take a child to a physician and the child looks like this, what the physician sees is this. So a physician will see a big signaling diagram. You can't see the details, but it doesn't matter because the physician might as well just see a bowl of soup. And the idea is that the traditional approach to medicine in the United States involves thinking of a child and thinking of a person as a series of chemical reactions. And if you add some chemical reactions, you go from this to this. As some drugs are there. The big picture though, and what motivates all of us here at WashU, is that the breakthrough therapies in the 21st century need to account for more. There's structure. And so if you bear with me for just two more slides of philosophy, we'll get into real science. What motivates us and inspires us is that we have this is that structural mechanics is really the next aspect to be added to the great continuum of how physics has advanced biology and medicine. Every major advance in medicine has come with an advance in physics. And so what Farid and Della and Elliot and I focus on is adding structure, the physics of structure and the physics of interactions to this large history of physics advances. So we have a center. So all four of us are pleased to be part of a massive 10-year, $50 million investment by the United States government, centered here at Washington University and at the University of Pennsylvania. And in the first half of the center, we asked three basic questions. The three basic questions that we'll talk about today are: how do cells know when fibrosis is around? How do cells adapt to that? And then how does that memory of fibrosis get locked into a cell? All right, so enough philosophy. Let's get on to politics. Politics. So we all have a. I'm speaking to people who could teach me much about how the heart works. The heart is supposed to have, as you guys know much better than I, that two major classes of cells: the cardiomyocytes, which are thought of as electrophysiologically active. They conduct, they contract. The fibroblasts that regulate and support the tissue, and they secrete some soluble factors and maintain the collagen. It turns out, as people in the Corey lab have shown very elegantly, this is overly simplified, and they interact. And one of the questions that motivates us is the following: so you can improve contraction, and you can improve the heart by exercise. And if you bear with me here, think about exercise. In exercise, what happens? Your heart rate goes up, your blood pressure goes up, you start sweating, and it's good for you. Your heart gets stronger. But now think about this here. When you see this person, what happens? Your heart rate goes up, your blood pressure goes up, you start sweating, but it doesn't help your heart. What happens is that your heart, instead of becoming stronger, starts to look more like this. These fibroblasts, for reasons we don't fully know, start to turn into what's called the myofibroblast, a larger contractile phenotype that produces collagen. Same type of stimulus, but a very different response. And so our group has a large multifaceted effort to understand how it is that this change occurs. You're going to hear about that from Farid and Della today, about how this change occurs. You're also going to hear from them how the mechanical aspects change the response of these tissues. But what I want to focus about now is the electrophysiological effects of how these myofibroblasts interact with the cardiomyocytes. And the last piece of introduction here is I want to describe the system that we work in. The systems that we work in, they're based off of very simple mechanical tests. So this is a device invented in Elliot Elson's lab 3 decades ago, but it's been a workhorse of the tissue engineering world ever since. This is actually the original one here, so you can see particle board and cheap materials. But the idea is that these artificial heart tissues were first invented in the laboratory of Elliot Elson. Thomas Eschenhagen made the first artificial heart tissue in the 1990s, and the basic components are cardiomyocytes. We typically use chicken cardiomyocytes, or now these days we use induced pluripotent stem cells. And if you mix those in with some fibroblasts and some collagen, you get these tissues that beat spontaneously, and we can perform experiments on them and drop drugs in on them as we do these experiments. And these teach us a lot. And I want to just show you, only going to show you a handful of experiments, mostly modeling that we'll talk about. But the one experiment I want to show, this is something that we published several years back, is an interesting experiment. And so to understand how myofibroblasts and fibroblasts interact with one another, we did the following experiment. We made tissue constructs, one with as few one with as few fibroblasts as we could put in, and one with extra fibroblasts. So if you look after 4 days of culture, and you can see these move here, you can look at 4 days of culture, you can see that the tissue constructs with extra myofibroblast beat harder. So with extra fibroblasts, the tissues are able to beat harder than the ones that have as few fibroblasts as possible in them. Look 2 days later, and the ones with more myofibroblast, they both have a higher baseline tension, but the ones with more myofibroblasts don't beat at all. And now the twitch force for the ones with no myofibroblasts is approaching that of the initial ones. And so you're going to hear about all kinds of mechanical interactions that occur. And Farid Al-Safai is probably the world's leading expert on the mechanics of it. And you'll hear from him today. So I'm not going to talk about that. You'll hear from Delaram and Elliot about how these cells interact with one another. What I will focus on is electrical aspects. And so I want to start off with some modeling studies. So we can now make very nice mathematical models of these interactions here. We take a piece of fibrotic myocardium. We look at how many fibroblasts and how many cardiomyocytes are there, and we put them in this idealized structure. The big red bricks are cardiomyocytes. The little yellow dots are cardiac fibroblasts, which have turned to the myofibroblast phenotype. So that we have very good models of the cardiomyocytes. Most of them have come from Washington University, from Norm Rudy, who's here at Washington University. But then these small fibroblasts, they'll connect to them and connect one another. And so the idea is that there are a couple of very nice models out there. One from the group of Wayne Giles and one from Sachs and co-workers. And the idea is that I'm sorry this doesn't appear to be very legible on Zoom. The idea is that we keep track of the intracellular voltage in each individual cell, and then note that we have to keep track of all ions. Ions that move out of one cell move into their neighbors. So if you're not a modeler, this equation might look intimidating, but it's really very easy. So what I want you to note is that there's a voltage in the cardiomyocytes, the Cs, and a voltage in the myofibroblasts. And so these two cell types, they have a capacitance. There's a certain amount that they store in the membrane. Then there are a whole bunch of ionic currents that go through. And then there's the passive flow. The passive part is a function of the difference in voltage between the two. And these have different resting potentials. Because they have different resting potentials, there's always going to be a tendency for them to talk to each other electrically. Now, because we have very limited time, although I'm very happy to discuss more in questions and answers, I want to talk just quickly about a couple of models we've developed and just show three interesting results. So the idea is that we couple existing models of contraction with existing models of the ionic currents here. We add two things to it. The first thing we do is we add a term that is dependent on mechanics. So the idea is that as cardiomyocytes stretch, they stretch the myofibroblasts. And so what we've added here, the thing that's new here, this is one of the many ionic currents that show up that go into the myofibroblasts. We note that the resistance, the tendency for there to be flow through these ion channels increases with stress for these. That's number one, and I want to show you that's an important factor. I want to show you the second thing I want to show is that there is a propensity there's a propensity for myofibroblasts to produce TGF beta, amongst other things. So TGF beta actually has direct interactions on the cardiomyocytes. They do two things. Number one, they change the density of T tubules, so they change the calcium handling. And number two is that they change the density of an ion channel called ITO. This is the transient outward current of potassium. And I want to show you two interesting things that show up here. So first, I wanted to show you what happens. So we modified these models. So the Sachs model and Meccano models. These shows how the myofibroblast changes its voltage as a function of time when stimulated. So there's an active component here, which is captured by Meccano and by Sachs. It turns out by adding in this mechanical component, we're able to replicate experimental data of Vasquez and coworkers, the classic data. more accurately. So we're able to recover the correct shape this way. Because we're able to add in a mechanosensitive ion channel and recover the shape, we think we're doing better. And we'll get to what this means in a moment. So this is now adjusting the mechanical part. The second thing we can do is that now, once we know that we have the right electric response for the fibroblasts, we can start looking at how different types of cells interact with each other. So point number 2 is that we think this ion channel, ITO, has an important effect. And so this important effect, it's something very interesting shows up here. So we have on the left and the right, these are all simulations. These are simulations of what we see in the myocyte and in the fibroblast. And it's very interesting. So on the left is human, on the right is cardiomyocyte. And so if you look at on the left is human, on the right is rat. So over here we have you see that as you add fibroblasts in a rat, the action potential gets slower. As you add fibroblasts in a human, they get longer. And I'm missing a slide, sorry. It turns out if we could show then now that this druggable channel, we can add biochemical inhibitors to ITO and eliminate this effect. And then mathematically, we can switch the ion channels between the two models and recover. So we know that one channel affects whether or not the action potential gets lengthened or shortened in response to fibroblasts. And so then the final thing we did is we asked, well, given that TGF beta affects this channel, the ion channel associated with ITO, what do the effects of TGF beta have on the action potential of the cardiomyocytes? So these are the experiments of Kauer, beautiful experiments in which cells in a dish have had varying amounts of TGF beta added. And you see the experiment, the more TGF beta, the longer the action potential. And so by adding 2 things to the model to account for TGF beta, we can identify that these 2 things that we added are adding T tubules. So TGF beta reduces the density of T tubules in cardiomyocytes, and it also reduces, which changes the calcium available from the sarcoplasmic reticulum, and also it changes the open probability of the channels because there are fewer channels. You see, we're able to capture these effects. And so these are simple examples of druggable effects at the material level. As in the last 4 minutes I have here, I'd like to talk about the next level up. So these are material effects that are druggable. The interesting thing is that there are passive effects at the tissue scale that are not druggable. It's very interesting. Not druggable by us. Smarter people like Vahid probably have good ideas of how to do it, but for us, we don't know how. So now we're taking out the active part. We're just using a passive model for fibroblasts, and we're asking what happens to the cardiomyocytes as a depolarization wave goes through. And there are 2 classes of things that happen. The first is, what we're seeing is voltage as a function of time. You can see where the myofibroblasts are because they have a different resting potential than the cardiomyocytes. So we're looking at now this depolarization pattern. You can see everything was started at the same time, but the cardiomyocytes slow down the conduction a little bit. And so cardiomyocytes always do this. The other feature I just want to very quickly show is that if we make a tissue construct like this with an island in it, you can see these effects. There are structural effects that go beyond the material effects. So now, if you go backwards here, we can see the island of myofibroblasts cause major disruptions. If this were asymmetrical, it might be possible to even start a reentrant arrhythmia. So very quickly in the last couple of minutes, if we do this for lots of tests, we find something very interesting happening. So right now so in continuum theory, myofibroblasts, it's the volume fraction, the fraction of the total volume that represents myofibroblasts that matters here. So in a numerical simulation, you can put this in. You can never measure this. So for the experiments, we're going to have a different x-axis. But what happens is that the more myofibroblasts you have, the more they get in the way of conduction. But then something interesting happens, which is that it drops down. Conduction goes to zero. It becomes absolutely impossible to conduct. But then later on, we get conduction. We get a little bit more conduction as conduction goes through the myofibroblast. So three things are happening here. We see that there's this passive region in which the myofibroblasts just get in the way of conduction. So conduction slows down. Then there's an active region here where very quickly the myofibroblasts collapse down, the conduction collapses down to zero, and we get this random excitation. There's no correlation between the stimulus and the response. And then if you have even more fibroblasts, then eventually we go to a passive percolation and we get a little bit more conduction back. Anyway, I realize I only have 1 minute left here, but I'm going to go 2 minutes anyway. So we've done this now in tissue constructs, using a calcium-sensitive dye. And you can see what happens in a highly fibrotic tissue. You can see that the calcium concentration here. Actually, excuse me, this is voltage. The voltage front breaks apart here, and so that you can see all kinds of crazy effects from these fibroblasts. If we do a series of these, this is something that was published several years back. We can see now this is a different x-axis than we had before. So now it's fraction of total cells that are myofibroblasts. We can't get total volume fraction. It's a hard thing to measure, but we see these same phenomena here in the experiments. We see that there's passive homogenization. It starts at this point, passive homogenization, and then regardless of how many extra myofibroblasts we have, we see a drop associated with this active percolation, and then we see a drop to outside of the homogenization bounds. These are limits that we can derive mathematically using statistical physics. So the big takeaway point, very big takeaway point, is we show that at the structural level, the myofibroblasts never help electrically. At the material level, we showed that they can sometimes make things faster. But at the structural level, we get percolation. We get electrical percolation, and then that can eventually lead to a second percolation threshold in which conduction stops entirely. I'm over time here. I apologize for going long, but there are two takeaway points here. So what we've seen is that there are material and structural effects that we can see in these tissue constructs that are electrophysiologic. The idea is that at the material level, we see that these interfaces between cell types can either extend or reduce the active potential. The effects can be minor, but we can reduce them, we can affect them using drugs. Then there's a second level, a structural level, where we don't know how to use pharmacological intervention yet, whereby myofibroblasts just interfere with depolarization and conduction. And then there are these percolation thresholds where the myofibroblasts start interacting with each other. That seems to be a key factor in the electrophysiologic side. So anyway, I'll close out here. And the US taxpayers have been very generous. Taxpayers in other countries have been very generous. But this, I will now close out and let my colleague, Dr. Ali Safai, who's a true world expert in understanding the mechanical effects, provide additional insight into very, very related phenomena. So thank you very much.
Thank you very much, Professor Guy, for your brilliant lecture. I have several questions about your lecture because we have short time and should schedule symposium to end at least 1.5. I just only ask one question. As an electrophysiologist, I definitely agree with you that in the heart, we are a balance between ionic channels, ionic currents, and passive properties such as fibroblasts and myofibroblasts. And I just question during pathology, you think that the balance between ionic channels and the fibroblasts, what happened? If it's going to the myofibroblast and the passive electrical property and passive mechanical property, why several great pharmaceutical companies work to ion channel blockers such as potassium blocker, amiodarone, lidocaine, and some other pharmaceutical and drugs for controlling arrhythmia and especially ventricular arrhythmia and ventricular fibrillation. If you are right that I believe in, we should control the arrhythmia with the modulation of the myofibroblast drugs, not ionic current. You know that during end of the ventricular tachycardy, we are going to the alternates. All the ionic channel becomes inactive because the potential goes to minus 60 or near the minus 55. In that case, the majority of the ionic channels becomes inactive. So the role of the myofibroblast or the fibroblast becoming more dominant than ionic channels. So we should control at the end of ventricular tachycardy, at the point of transition to the ventricular fibrillation, with some modulator of the myofibroblast or even fibroblasts, such as connexin enhancer or gap enhancer, such as rotigapide and some other connexin enhancer. What is your idea about it?
This is a brilliant idea, and of course, I'm thrilled to hear your ideas. It's again an honor to be having the chance to interact with the true world expert on cardiac electrophysiology. We've published some ideas on this, and actually in this paper that came out in Biophysical Journal in 2021, and I forgot to acknowledge the lead author, who's Fusheng Liu, a student who is in Xi'an, China. In that work, we looked through a whole family of ion channels in both the fibroblast and the cardiomyocyte, and it seems that for number one, the gap junctions don't seem to be a major player in the effects of myofibroblasts on cardiomyocytes. So the gap junctions are rarely, if ever, seen between cardiomyocytes and myofibroblasts in vivo, and then the key problem is the resting potential and the soluble factors. So in terms of soluble factors, it really seems like knocking down inflammatory cytokines, according to our model, which seems to be predictive of data. Knocking down inflammatory cytokines can have a substantial effect on these cardiomyopathies. And then in terms of in terms of potassium blockers being very effective, that seems to be consistent with the modeling. The potassium blockers affect the cardiomyocytes, but what they do is knock out ITO. And when you knock out that particular current, many of the effects of the myofibroblasts are eliminated, or you can even reverse them. So your insight is is spot on, very strong, and I think that this type of integrated modeling has potential for informing future therapies.
Okay, thank you. for your answering. And now I want to welcome to the Dr. Ali Murad, Dr. Farid, exactly. Dr. Farid, please introduce yourselves for the Iranian and also foreign country participants, and you can have the symposium. Welcome.
Thank you so much. سلام عرض بخیر خدمت همگی. فرید علی صفایی هستم، استاد دانشگاه نیوجرسی. قبل از اون دوره فوق دکترا دانشگاه یونیورسیتی پنسیلوانیا بودم و شانس این داشتم که همکاری کنم با دکتر گایجانی و الیوت السون و گروهشون در دانشگاه واشنگتن سن لوئیس. Good morning, everyone. It's absolutely a pleasure to be here with you, although virtually. My name is Farid. I'm a new faculty at the New Jersey Institute of Technology. And today I'm going to present some of the work I did during my postdoc in Shenoy Lab at the University of Pennsylvania. So let me, if I can share my screen with you.
شیر اسکرین رو میتونیم بزنیم. ما تصویر شما رو هم نداریم. اوکی. الان داریم، تصویر رو داریم. اسلایدتون رو داریم. اوکی.
تصویر من هستش؟
تصویر شما نیست. من نمیدونم چرا. شاید دوربین زوم تو روشن کن پلیز. آره
روشنه.
نه یه دو تا پایین چیز، هم یه دونه میکروفون داره، یه دونه ویدیو داره. ویدیو تو باید
روشن بودش.
آره یه لحظه من دیدم. بله ببینم. من شاید من ندارم. نه داره. از تصویر شما هست. بفرمایید. اوکی.
Again, good morning, everyone. As I mentioned, the topic of our work is mechanobiology. This is the work that I did during my postdoc at the University of Pennsylvania in close collaboration with amazing people at the University of Washington in St. Louis, specifically Dr. Janine Elliott and Dr. Shakiba. As I mentioned, the topic of our work is mechanobiology. And I believe that mechanobiology is a relatively new field for many of you. So I would like to start my presentation with a brief introduction to mechanobiology. And let me talk a little bit about why mechanics is essentially important in our health and also different diseases. I guess we all know Robert Hooke because he was the first to visualize microorganism, and he was the one who coined the term cell. Basically, Robert Hooke was the first one who discovered cells. But Robert Hooke is also a very well-known scientist in the field of mechanics and physics. My background is all in mechanics. And in mechanics, everyone knows Robert Hooke because in 1660, 5 years before, he discovered the most fundamental law of mechanics, which is called Hookean law nowadays. But I think even Robert Hooke himself did not expect to see any connection between his two discoveries, any connection between mechanics and biology. But after more than 3 centuries, we as mechanobiologists are now studying the interdependence of Hooke's discoveries, the interdependence of biology and mechanics, or in other words, how mechanical factors, for example, like stiffness, geometry, and force, impact different cellular behavior. For example, how force, geometry, and stiffness can impact proliferation of cells, can impact differentiation of cells, or even gene transcription. And I think probably the best example to describe mechanobiology is cell differentiation. we all know that in the body, we obviously have different organs. And each organ obviously has its own cells. For example, consider the brain. In the brain, we have neuron cells. In fat tissues, we have adipocyte cells, right? In bone tissues, we have osteocyte cells. In muscles, we have myocyte cells, and so on. And also, we know that all these different types of cells are formed by stem cells. The stem cells can renew themselves, or stem cells can differentiate and become neuron cells, or they can become osteocyte cells, they can become myocyte cells. So all different types of cells are formed by stem cells. But the important question, and what is really interesting for us, is that mechanics can be also an important factor in cell differentiation. How? So here I'm going to show you how mechanics can influence cell differentiation. Here in this example, we have stem cells cultured on hydrogels with different stiffness, but under exactly the same chemical environment. As you can see in the first case on the top, we have cells that are cultured on a very soft ECM and a very soft matrix that basically mimics the stiffness of the brain, like in the order of 0.5 kilopascal. In the second and third cases, you can see that cells are cultured on stiffer matrices with the stiffness of typical fat and muscles. And finally, you can see that in the last case, cells are cultured on a rigid substrate, very stiff substrate, which obviously represent the mechanical environment of bone tissues. So what's the results? Interestingly, you can see that cells on the softest matrix, they become neuron cells. Cells on the stiffer matrices become adipocyte and myocyte cells, and cells on the rigid substrate become osteocytes. So I guess this example clearly shows that the stiffness of the extracellular environment matters, and that stiffness can determine cell fate. And I have to tell you that it is not just stiffness. Other mechanical factors like geometry and force, they have been also shown to impact cell behavior. For example, in this example, we can see the effect of geometry. So if we change the cell geometry, and if we confine a cell to a very small area, or in other words, if we do not let the cell spread freely, so basically we confine the geometry, what will happen is that we will see that cell go through the process of apoptosis. Basically, cell will start to kill itself. But on the other hand, if we let the cells spread freely, we can see the growth and proliferation of cells. So this example again tells us that geometry can control cell life. And also, if we apply mechanical forces to a cell, for example, here we have a cell, and if we stretch a cell in one direction, as you can see, then we will see that many changes in the cell will happen, including, for example, changes in the cytoskeletal organization. Cells become stiffer, and then we will see changes in gene transcription. So cells under mechanical forces show different pattern in gene transcription. So the take-home message of this introduction is that mechanical signals like stiffness, geometry, and force, they all can change cell behavior. Okay. So with that short introduction, I'm going to briefly present some of the work I did on the mechanobiology of cells and tissues. And I'm going to start with extracellular forces, external forces. So we all know that cells in the body experience external forces that can be in the form of tension, compression, or shear. And we also know that these external forces sometimes can damage cells, sometimes can hurt. Like, for example, in the brain, when we have brain injury, mechanical forces are transmitted to the brain and also to the neuron cells. And during brain injuries and concussion, we know that these mechanical forces can be really harmful. Also, sometimes we know that these mechanical forces can have positive impacts. For example, consider cyclic forces during physical exercises on bone tissues and muscle tissues. They can help us to have healthier tissues. So a great example of cells under external forces is the human ocular surface. This is the work that we did 2 years ago in collaboration with Dan Ha at University of Pennsylvania. So I guess ocular surface is a great example where we can study the effect of mechanical forces on cell behavior. So as you may be able to appreciate from this slide is that each time we blink, cells on the ocular surface experience external forces. But what is important is the fact that these external forces are really necessary for the normal functions of cells. So, which means that any alterations in these physical forces, for example, because of diseases such as dry eye disease, can lead to abnormal cell behavior and inflammatory responses. So basically the question is that what will happen if we don't blink, if we don't apply these mechanical forces to these cells on the top of ocular surface? Or if we have, for example, a disease like dry eye disease, and then we don't have enough lubrication, we don't have lipid layer in our tear film. So the nature of mechanical forces will change. So what will happen to cells on the top of ocular surface? And that's what we decided to study. So in order to study that type of diseases like dry eye disease, there is obviously great interest in making artificial tear films that can restore the original force field in patients with dry eye disease. So to this end, obviously, eye-on-a-chip system can be a good platform to test the effects of different drugs. Instead of testing on human beings, on animals, we can come up with organ-on-a-chip and then try and then test the effect of different drugs on the organ-on-a-chip systems. But one of the first steps in designing such system is to ensure that cells in our system experience the same level of physical stress as cells experience in the human eye. So basically, we have to make sure that the system that we have is really similar to the human eye. So, and that is exactly where we need a theoretical model. So without going into details and without going into the mathematical details, we develop models for the ocular surface, for the tear film, and also for the eyelid. And then by coupling all these models together, we could determine the complex physical stress field at the ocular surface, on stress field of the ocular surface, including the pressure and shear stress that cells experience during blinking. Basically, what we did, we determined how much mechanical forces do we have on our cells on the ocular surface each time that we blink. And based on that calculation, we could develop eye-on-a-chip system. And with that eye-on-a-chip system, we can make sure that the system can faithfully recapitulate the biomechanical environment of the human ocular surface. Okay, so after talking about external forces, I would like to talk also briefly about internal forces. So we know that cells in the body not only experience external forces, but also generate forces on their own. So, and I think one of the earliest pieces of evidence was presented in this science paper in 1980. And that in that paper, the author shows that cells cultured on a soft polymer would pull on the polymer and wrinkle the surface. So what does it mean? This means that cells can generate forces on their own, and they can transmit these forces to their surrounding environment. And now, after about more than 2 decades, we know that these internal forces are really essential for many biological processes. For example, if we think of a cell as a car, then these forces are the engine. Cells use these forces to migrate. Cells use these forces to invade to the other organs. And Also, another important factor that I think Dr. Elliot Elson and Dr. Shakiba are going to talk about that is the role of these mechanical forces in the process of, for example, wound healing, and also in the process of some diseases like fibrocontractile diseases, including fibrosis. We know that cells use these forces to close the wound, right? During the process of wound healing, normal fibroblast cells become myofibroblast cells, they become hyperactivated, more contractile, they generate higher forces. Then these cells migrate toward the wound site, they start to deposit collagen fibers at the wound sites, and then by generating high contractile forces, they try to close the wound. That's the process of wound healing. But obviously, after the closure of the wound, if this force generation continues and if cells do not return back to their original state, and if they stay and remain hypercontractile, then obviously we will have formation of scar. And even in more serious cases, that can lead to formation of fibrotic tissues. So internal forces can have both positive and negative roles in the body. Positive like wound healing, negative like formation of fibrosis. And of course, I think the worst negative effect of cellular forces, internal cellular forces, is their involvement in cancer progression. Cancer cells use these forces to escape from the primary tumor and invade other organs. So it is really important to measure these forces. So the key question is that how much force a cancer cell can generate. So, and here is the specific question that we wanted to answer. We wanted to, by measuring cellular forces, we wanted to answer that how much force a cancer cell can generate. And if we have a cancer cell in a normal body, or if we have the same cancer cell in a person with diabetes, in older people, where we have different mechanical environment, how much that mechanical forces will change. So, and then we know that if we think of the cell as a car, then these forces are the engine. So by measuring forces, we can understand whether cancer cells are become more aggressive and invasive or not. So this is exactly what we did in 2016 in collaboration with Mingming Wu at Cornell University. So we know that the physical properties of tissue change during aging or after injuries or diseases such as fibrosis. We know that in all cases, the tissue becomes stiffer. So the question is how these changes impact the behavior of cancer cells. Do cancer cells become more aggressive or do they become less invasive? So to answer this question, we need to, obviously, again, as I mentioned, we need to measure cellular forces. But how we can do that? So here is the process that we did. First, we need to culture cells in 3D collagen network, 3D type 1 collagen networks. And as you may see, we have small fluorescent beads. As you can see, this is a small fluorescent beads. We have a small beads, and these beads basically are covalently attached to collagen fibers. So if collagen fibers move, these microbeads also will move with the collagen. So by tracking these beads, we can determine the displacement field in the collagen matrix generated by the cell. So if in the second panel, you can see the displacement field generated by a MDA breast cancer cells. So we have the displacement field. Okay, then how we can measure mechanical forces, how we can measure how much force it has been generated. So to measure force, we need to develop a theoretical model that basically gets the displacement field from experiments as an input and then calculate force as an output. And this is what we did in these two papers. So without going into details, we developed such a theoretical model, and we use this model to measure cellular forces generated by MDA breast cancer cells. As you can see, we have an array of 9 collagen matrices. These 9 collagen matrices have been made with different collagen concentration, different temperatures of polymerization, and also different levels of cross-linking. So as you can see, all of them have different microstructures. And by measuring their stiffness, we also found that they have different stiffness. Some of them are very soft. Some of them have the stiffness of a normal tissue. And some of them are very stiff and show the stiffness of, for example, aged tissue, the stiffness of older people, or tissue after injury, or tissue with, and also as you may see in some of them, these collagen matrices, some of them have high level of glycation. So we just wanted to have these collagen networks to represent tissues in a person, for example, with diabetes, when we have higher level of sugar. So we have different collagen structures. Some of them are still very stiff, some of them have higher level of glycation. So, and then we use the theoretical matrix model and we measure cellular forces in each of these matrices. And then, as you can see, we plot the force here in the y-axis. We plot the force as a function of collagen stiffness in the x-axis and cellular forces in the y-axis. And I think what we can appreciate from this plot is that we found that cancer cells generate higher forces in stiffer collagen matrices. And why it is important? So first of all, we know that collagen stiffness increases after injury. So when you have injury, when you have a wound, after process of wound healing, collagen stiffness increases. Also, we know that during aging, collagen becomes stiffer and stiffer. So we have stiffer tissue. Also, we know that with diseases such as fibrosis or diabetes, the collagen becomes stiffer and stiffer. So now you can see that cancer cells becomes more aggressive in people with previous injuries. They become more aggressive in older people. They become more aggressive in people with diseases such as fibrosis and diabetes. And I guess that can explain partially, not everything is mechanics, but that can partially explain why, for example, chronic wounds, aging, and diseases such as fibrosis and diabetes can increase the risk of cancer invasion. So it means that if you have a cancer cell in a normal body, or you have the same cancer cell, put it in a person which is older, or put it in the body after the process of wound healing, cancer cells will migrate and will invade faster and more aggressive in these environments. So, and then finally, as my last example, I'm going to talk a little bit about the interdependence of internal forces and external forces. So I reviewed external forces, then internal forces, and then how these two work together, internal forces and external forces. For example, consider fibroblast cells in connective tissues. We already talked about internal forces, and we mentioned that fibroblasts use these internal forces to close the wound during the process of wound healing, right? We also mentioned that these internal forces can be the main cause of and the main source of fibrosis. So on the other hand, we know that fibroblasts in the body can experience external forces during daily physical activities, right? For example, when we sleep, these cells experience compression. When you sleep on your back, the cells in fibroblast cells in connective tissues experience compression, mechanical forces in the order of kilopascal. So the question is that how these compressive forces affect cell behavior and cell internal forces. So to answer this question, we culture a fibroblast on a rigid substrate, and then we put a plate on the top of the cell and apply compressive forces. But before showing the results, let me first talk a little bit about the cell cytoskeleton, because we want to see what will happen to the cytoskeleton of cell after cells experience compressive forces. As you may know, the cell cytoskeleton is composed of several components. The first element is actin that can polymerize or depolymerize continuously. The second component is myosin that can attach to actin and generate force dipoles, and that's how basically cells can contract. Then the next example is microtubules that usually experience compression as the cell contracts. And the last component of the cell cytoskeleton is intermediate filament, and specifically in fibroblast vimentin intermediate filament. So now let's get back to our problem and see what will happen to the cytoskeleton of fibroblast cell when the cell experiences compressive forces. We can see that under compression, actin filaments are depolymerized and the level of myosin phosphorylation significantly decreases. So which means that under compression, when you sleep on your back and cells experience compression, cells become low contractile and generate less internal forces. So if you have a wound there and you want to, fibroblast cells help to close the wound, putting compression on that definitely is not going to help. So we know that cells become less contractile under compression. So what is the consequence of this reduction in actin-myosin contractility, specifically, do we have any change in gene transcription or not? So we can see that under compression, histone deacetylase or HDAC, we can see that HDAC translocates from the cytoplasm to the nucleus. So cells under compression have a higher level of HDAC inside the nucleus. And we know that the role of HDAC will condense chromatin. And that's exactly what we see in this slide. Cells under compression have higher chromatin condensation and they become transcriptionally less active. So it means that when we put compression on fibroblast cells, they become transcriptionally less active. But interestingly, when we remove the load, we can see that cells get back to their original condition and they will have, again, their normal gene transcription pattern. So it tells us that as long as we are sleeping and compressing fibroblasts, these cells cannot have their normal gene transcription pattern. But immediately when we release the load, they get back to normal condition. So obviously an interesting question that we hope that in future we can work on that. The question is that what will happen if we keep these forces for a very long time? For example, in a person with a severe disability that cannot move or roll over, and then we have permanent compressive forces on the connective tissues and fibroblasts, then what will happen? So with that, I finished my presentation. I would like to thank again my collaborators, especially Dr. Guy Jennings, Dr. Elliot Ellison, and Dr. Shakiba at Washington University, in St. Louis. Also, I would like to thank my other collaborators at the University of Pennsylvania, at Stanford, at ETH, and also Cornell University. So please feel free to contact me if you have any questions or suggestions. You can see my email address here, also my Twitter address. So thank you again very much for your attention, and I hope you have a nice rest of the day. Thank you.
Thank you, Dr. Farid, for your brilliant presentation. I
appreciate that.
Very enjoyed all of them, and I have several questions, but I don't think that we don't have extra time. But I should ask a question. At first, I thought that the Washington University wanted to destroy all the pharmaceutical companies because Dr. Guy destroyed the pharmaceutical company anti-HIV drug. You destroyed all the pharmaceutical company focused on the eye drop, and because you all of your results show that we can treat dry eye with the pressure beside of the place or eye drops that, and several other diseases such as cancer or metastasis dependent to the pressure to the medicine or pharmaceutical drugs. So I am okay. Maybe during the pathology or physiology is okay. But the major question is if your mechanobiology is very important in treatment of disease. Okay. In near future, we should massage the body and inject some anti-cancer drug? You mean that
So
we can
that's absolutely good question. With respect to cancer cells, obviously different cancer cell types may have different behavior, but specifically for breast cancer cell, it has been shown that applying compressive forces would be really
yeah.
bad. So if you have breast cancer cells, obviously the last stage of detecting cancer cells is just by touching. You will have stiffer tissue. Just by touching the tissue, we realize that it's stiffer. And the reason it's stiffer is because of three things. First, cancer cells produce more cross-link, so the collagen becomes stiffer. They are more contractile, they are stronger, they generate more forces. So we have collagen realignment. That's the second contribution for the stiffness. And the third case is proliferation of cancer cells. They produce more and more. So in a small and limited volume, we have a lot of cancer cells. And these three combined together will lead to, when we touch the tissue, we'll realize that we have a stiffer tissue. So with respect to your question, if we apply mechanical forces to cancer cells by massaging, what will happen? Compression, massaging means shear forces, like when we do like that. But compressing that, it has been shown that if you compress breast cancer cells, they become more invasive. And what is the mechanism is not clearly known, but they suspect that the nucleus plays an important role in that. So by applying compression, you generate tension in the cell membrane. What will happen? We have mechanosensitive ion channels in the membrane. They will open, and then ion can easily transport in and out. For example, calcium can flux into the cell cytoskeleton. Now, if we have more calcium, which means that we have calcium pathway activated, we have Rho and ROG pathway activated, and these are the pathways that, like, they are the engine of the cells. So we are activated this engine. So cells get more energy and they can move faster. So compression is not really the best way to treat with cancer cells. But so we are saying that applying compression makes cells becomes more contractile. But when we have a wound or when we have injury, and after injury, we want to have the best process of wound healing, then so we need cells become more contractile, right? Then mechanical forces can help us. So because again, cells become more contractile, they migrate faster, they get to the wound site faster, they can generate higher forces, and they can close the wound. So it means that it has, as I mentioned, it has both negative and positive effects. It depends in which context we are studying them.
And thank you so much, Dr. Farid. And also,
my pleasure.
I have one more question. If we are going to the end of the disease, not cancer, for example, arrhythmia or myocardial infarction, we going from the healthy people, healthy tissue, to the end of living. The effect of the mechanobiology, you think that become greater or lesser? You mean that
obviously the answer is greater. There is a new paper published a few months ago in Finland in Science. They showed that let's say one cell that produce hair, they pick the stem cells from a person which is really old, with age of 80. They take out that stem cells and they put it in the tissue of a young person, and the stem cells start producing hair again. So which means that the stem cells are not getting old. The environment around the cells are getting old. So it means that as we get older and older, during the process of aging, the physical property of the around cells will change, and that physical property will affect cell behavior. So if you can rejuvenate the physical property of the around cells, cells are still young. So stem cells in an old tissue, when you put it in a young tissue, it works and it'll have normal function. So your answer is that as we get older and older, so we see more changes in physical properties, we get more stiffening, and then obviously the effect of mechanics and mechanobiology becomes more prominent.
Thank you so much. But in that case, we should think about the CPR, about the cardiac massage and defibrillation. I think that if is right, we should completely change the guideline for the CPR and use more time focus on the cardiac massage than defibrillator. Yeah, thank you so much, Dr. Farid.
Thank you, my pleasure.
I welcome and invited the third person. I don't know, Dr. Elliot and Dr. Shakiba Delaram is connected to us or not. خانم دکتر دلارام شکیبا دانشجوی پزشکی دانشگاه گلستان و دانشجوی خود من بود در دانشگاه علوم پزشکی گلستان دانشکده پزشکی. بعد از اینکه پزشکیشون رو تموم کردن مستقیما رفتن دانشگاه واشنگتن و پستاکتشون رو در واقع با سوپروایزری آقای پروفسور السوم و پروفسور گایجنین روی بحث در واقع تبدیل فیبروبلاست به مایوفیبروبلاست و پدیده پورترشن گرفتن که یکی از قوی‌ترین ژورنال‌های ریمودلینگ فیبروبلاست و مایوفیبروبلاست در این زمینه بود. الان هم در رشته تخصص پاتولوژی دارن درسشون رو ادامه میدن و یکی از افتخاراتمون مثل آقای دکتر فرید و سایر عزیزان هستن در واقع بحث فیبروبلاست در دنیا و یا هم بحث مکانیکال پرشر. میشه لطفاً فیلمشون رو بذارید.
Good afternoon, everyone. Thank you for having me. I'm very excited to be here virtually with you at Physiology and Pharmacology conference. I'm delighted to present our study on mechanobiology of fibrosis. This has been a work that I've started during my postdoc at Washington University in St. Louis, and it's still an ongoing project now that I am a physician resident in pathology department at St. Louis University. Hope we can get the conversation going here and find some common ground. Some of the results I'm presenting here hasn't been published yet, so I would really appreciate your feedback. Let me start with what inspired me to work on mechanobiology of cells and tissues. I'm interested in cell matrix interaction in 3D collagen matrices and how these interactions on one hand can lead to healing wounds, but on the other hand can form fibrosis, and also it can play a very important role in tumor progression. Fibrosis is a serious health problem, and it's characterized by excess accumulation of extracellular matrix that can occur all over the body, as you can see in this slide, and can actually progress to organ malfunction. Fibrotic diseases include a wide spectrum of clinical entities of unknown etiology, for example, idiopathic pulmonary fibrosis. We call them idiopathic because we don't fully understand how it occurs or what are the processes that accelerate it or can slow it down. And there is still no definitive treatment available for it. Why is that? Because it's not a disease of pure chemistry. There's a lot of physics going on. Let me show you a video I took tracking a fibroblast for about 2 days in 3D collagen matrix. Look how these cells interact and remove and the matrix around it. And this highly dynamic interaction is driven by physical forces generated by a unique cytoskeleton system within the cell. And it's absolutely essential to a variety of biological processes such as morphogenesis, wound healing, cancer metastasis, and immune response. So these physical forces are important to study to understand the question of how cells can change the architecture and function of tissue and organ just by their interaction with the extracellular matrix. The question of how cells feel, how they remember, and the one that I consider the most important is how they can adapt the collagen and their microenvironment, which is called ECM remodeling. So I'm giving you a more visible example of fibrosis, which is Dupuytren's contracture. It's a hand deformity caused by excessive pulmonary fibrosis that can lead to finger contracture, as you can see it right here, and can cause by contractile myofibroblast under the skin. These are well-differentiated cells, essential for wound healing, that are responsible for tissue remodeling. But this is what happens when they get hyperactivated, where they shouldn't have. And you can see here that quiescent myofibroblast, actually fibroblast, in normal hand can turn into hyperactivated myofibroblast, aligning the matrix around them and forming these fibrotic traction fibers. So our focus is to look into this remodeling and this transition, and to be able to stop this, we are asking these questions. What are the mechanisms that cells use to interact with the extracellular matrix? What are the mechanisms of fibroblast to myofibroblast transformation, and can we stop this? So this is an outline of my journey. So we started with some fundamental questions on what the mechanisms is behind the fibrosis. To answer this question, we first started to look at fibrosis at tissue levels, then at individual cellular level, and then even smaller at subcellular levels. And these basic science knowledge at different levels actually helped us to come up with a few ideas on how we can stop and possibly prevent the formation of fibrotic tissues. And now, and our ongoing project is how we are planning to use this knowledge in clinic. So first step, we started with a simple model to study the cell matrix interaction. This is a picture of our cells in a 3D collagen matrix. I made these tissue constructs with human dermal fibroblast immersed in rat tail collagen type 1, which is in green here, and cells are in yellow. And I did the live cell imaging and followed each single cell from the very beginning under confocal microscope as they remodel the extracellular matrix around them over time. And then we analyzed the volumetric imaging stacks, as you can see right here, for each individual cell. So what do we know about the remodeling, and what do we not know? We know that remodeling extends over many cell lengths and can shrink the tissue down by communicating over very long distances. But what is not known, however, is what happens locally in the close proximity of each cell, how cells start interacting with the collagen. So first asked how isolated human dermal fibroblasts adapt their morphology and structure and their microenvironment in response to being embedded in a collagen gel from the very beginning. As expected, cells were initially round and they periodically extend and retract very small processes, and as these extensions and retractions progress, the collagen would be drawn in and accumulate in the vicinity of the cell. So we developed a computer code to measure changes in collagen density around the cell over time, and this graph is actually what we observed at level of cell processes. We estimated the density of collagen around the cell over time at several distances away from the cell, and as you can see, there is a significant increase in collagen concentration as a function of time, and actually it somehow can reach a steady state over time scales of hundreds of minutes, and they kind of stabilize. And at each time point, you can see most of the collagen accumulation is around the immediate vicinity of the cell, and it decreases as you go further from the cell. And these different colors represent different distances away from the cell, and again the big change in the beginning shows the importance of the first several hours of cell ECM interaction that we mainly focused on that. So then we looked even closer at each cell, and we noticed the significant portion of this accumulation happens around these little protrusions, which indicates that protrusions plays a key role in accumulating collagen at early stages. Here you can see an SHG signal of cells and collagen separately, and the combined image here. And as you can see, just even very small protrusions can accumulate more collagen than the cell body. Then we asked, what are the main cellular components responsible for the formation of the cellular protrusions? And to answer this question, we disrupt different components of cytoskeleton, including microtubules and actin filaments. As you can see, this is a control cell, and this is when we disrupt microtubules with nacatazole in our tissue constructs, and this is where we disrupt actin filaments by treatment of the tissue with cytoclasine D. This is a 3D construct of a tissue with cells treated with nacatazole, so their microtubules are depolymerized, and we found that with depolymerization of microtubules, cells lose their processes and they become round, as you can see when you compare them with control condition here. However, they're still contractile and they can remodel the matrix a little bit around them, as you can see here, but obviously not as efficient as when they develop protrusions like this. So lack of protrusions due to disruption of core microtubules leads to a reduction in tension at the cell matrix interface. So this shows the importance of cell protrusions in accumulating collagen and generating tension. Next, we pre-treated fibroblasts with cytoclasine D, which is an inhibitor of actin polymerization, to study how cell matrix interaction is affected by disruption of actomizing contractility. And as expected, no collagen accumulation was observed around the cytoclasine D treated fibroblast. On the other hand, cell protrusions, as you can see them here, grew in both length and number, which is very interesting. This indicates that formation of protrusion is regulated by a mechanical force balance between actomizing contractility and microtubule extensions. Without microtubules, as you can see here, there is no protrusions. But as tension in the cytoskeleton decreases by disruption of actin, microtubules experience lower compressive forces and can grow in length and number. Example of how lowering the actomizing contractility with different drugs like cytoclasine D and Y27632 treatment, which both decrease the contractility of the cell, can lead to lengthening of the protrusion by unrestricted microtubule polymerization. The green in both is microtubule, in red is actin. Here's then a schematic summarizing these findings. First, cell protrusions play the key role in accumulating collagen, and these protrusions contain both microtubules and actin filaments, and actin filaments experiencing these contractile forces and therefore generate this compression on top of the microtubule, and these compressive forces can in turn restrict the excessive growth of microtubules. So we conclude that there is a balance between actin contractile force and microtubule growth. So if it disrupts these compressive forces by depolymerizing actin, then microtubules are experiencing less compression and they can grow excessively in both length and number. Cells that are unable to form protrusions, the nocardosol pretreated cells, accumulating significantly less collagen around themselves and less remodeling. So now that we know how important those are in the process of remodeling and occurrence of fibrosis, we were wondering, can microtubule disruption stop progression of fibrosis? Well, it's not that simple. As you can see here, disruption of microtubules before embedding the cell within the matrix significantly reduce collagen alignment compared to control, because cells lose their protrusions. If you compare these two, you can see significant collagen alignment here versus the collagen, the nocardosol treated cells. But if you wait until the cells are already adapted to their environment and then disrupt the microtubules, it results in retraction of that protrusion that has been formed and result in higher collagen alignment. So the main message here is we found that fibroblasts use completely different mechanisms in early, which I think the most treatable stage of these interactions, and that their responses to drug can be the opposite of what they would be in the later stage. So by now we hypothesize that there is this two-way feedback between protrusion and collagen that help the tissue become fibrotic. So on one hand, these cell protrusions interact with the matrix and remodel and align the collagen fibers in their vicinity, and on the other hand, these aligned fibers also stabilize this protrusion and help them to promote the growth of the protrusion in the direction of collagen alignment. So we think that there is this two-way feedback between collagen alignment and protrusion stabilization that enables fibroblasts to become spread, polarized, and contractile over time. So at the end, they would be able to form these traction fibers in between to form fibrosis. Then we ask, how the protrusions does this, what is the mechanism, and we've measured the strain generated around the cell protrusions using this strain mapping technique, and as you can see there's this spectrum. As you go throughout the red, it's more tension, and blue is compression. And what we expected to see first was when you put in a cell in a uniform collagen matrix, the cell would draw collagen in at the end using processes like this, and after a while you got like concentration of collagen around the cell. So we expected to see most of the tension here at the tip, but what we saw, as you can see, is totally different. So you can see that there is compression at the tip and a gradient of tension laterally to the cell process. And then when we went back and checked the actual microscopic images, these two are 3D constructed construction of the same cell at two different time points that we mapped for strain, we realize that at this stage, this protrusion is growing between these two time points. So it compressed the matrix while growing and at the same time pulling on the collagen and generating tension laterally to the protrusion. So this is another example in this picture of how collagen would draw in and would accumulate laterally to the cell processes, this is exactly where the cells exert the most tension. And if you look closer, Dr. Khoury, for the invitation. Thank you. And I hope the internet connection would cooperate and we can chat. But if not, I am providing you with my email. I would appreciate your feedbacks or comments, or I can take your questions. Thank you very much again, and have a great rest of the day. Thank you.
Okay, I welcome to the Professor Elliot Elson for joining us. Professor Elliot.
Easy.
Nice to hear and see you, Elliot.
Very glad to be here.
Yeah, thank you so much. I know that it's very hard for you and your team because you are the early morning, and sorry for that.
No, it's because I'm a complete idiot about trying to use Zoom. That's why.
Yeah,
the morning is fine.
Yeah. You are the last speaker, Professor, and you have time to maximum 32 minutes.
Okay.
Just at first, I introduce you for my colleagues and the participants.
Okay.
And you can free on stuff. Okay. Let me introduce one of my best colleagues and friends, Professor Elliot Elson, one of the greatest scientists in the field of the tissue culture. Professor Elliot Elson, and Emeritus Professor in the Department of the Biochemistry and Molecular Biology at Washington University of School of Medicine, St. Louis. And recently, I don't know exactly when, is elected to the National Academy of Sciences. It's a very great position. I congratulate to you, Professor. Elson is widely known for his role in the pioneering microscopy method that have become the standard tools in the field of the biophysics. These methods, called fluorescence correlation spectroscopy and fluorescence recovery after photobleachings, are used by thousands of the researchers around the world to study the complex inner working of the cell and the environment. His early papers on these techniques have been cited in the scientific literature more than 5,000 times, highlighting their impact on the field. Professor Elson completed his bachelor and MSc from Harvard University, went to earn doctorate degree in the biochemistry from Stanford University in 1966. He continued training as the postdoc researcher at University of California, San Diego, before joining the faculty of the Cornell University in 1968. He moved to Washington University School of Medicine in 1979, joining the faculty of the Department of the Biological Chemistry, and he's working to the Iranian and love the Iran and Iranian researcher and people and students, and his labs is one of the most favorite labs in the Washington and everywhere. To welcome to all our university lecturers in Iran and all the students from Iran and everywhere. to love to work in the line of the major subject of the Professor Edson. Professor Edson, I welcome again to you and thank you to accept our invitation to the 25th Physiology and Pharmacology Iranian Congress, and you are unpassionately patient. You speak about a biomechanical model for transformation of the fibroblast to myofibroblasts. Is all. Yes.
Thank you very much, Vahid. My next job is going to be to get the share screen part working. And I think, let's see if this does it. So here I've got the PowerPoint.
We don't have your screen. Oh, yes, we have.
Okay. Okay, now let's see.
We have your picture and your sound. Go ahead.
Okay, thank you very much. I'm going to be talking about this cluster model for the transformation of fibroblasts to myofibroblasts that I've been working on for some time. And as first of all, let me thank Professor Corey for inviting me to participate in this conference. And let me say that I'm happy to do so, but only sorry that I have to do it by this Zoom. I would rather be with all the rest of the conferees in Iran. That would be more fun, and I hope I can get there and talk to those of you who are interested in this sort of thing at a later time. So I'm going to begin by giving a kind of preface to what I intend to talk about. And as the title says, we're going to be talking about how cells maintain their biomechanical homeostatic steady state. And so let's begin by pointing out something that you all already know very well, which is that cells are embedded in a viscoelastic extracellular matrix composed mostly of collagen and also other proteins and proteoglycans. Now, the cells are attached to this extracellular matrix by surface integrins that connect the internal actin-myosin cytoskeleton across the cell membrane to the extracellular matrix. And therefore, when the cells are able to contract their actin-myosin cytoskeleton, as Dr. Shakiba has shown you, they can compress the collagen, and that's one way in which fibroblasts and myofibroblasts can adjust the biomechanical properties of the matrix. So why is this important? Well, it's important because the biochemical and biomechanical properties of the matrix determine the differentiated form and function of the cells that are embedded within it. And I'm going to talk a little more about that in just a few minutes. And since the matrix controls how the cells develop and how they function, it's very important that the matrix be maintained in a steady state, we call it a homeostatic steady state, so that the cells will be able to continue functioning as they are meant to do. So let me go to the next step of this, which is that to maintain this steady state is in fact the task of the fibroblasts, the connective tissue cells that are in the extracellular environment, the extracellular matrix of all solid tissues. So the fibroblasts, by exerting small contractile forces and by secreting matrix components such as collagen and other proteins and other kinds of molecules, that the fibroblasts can adjust slightly the properties of the matrix to maintain the proper mechanical environment for the function of the cells. On the other hand, when there's a very serious perturbation of the properties of the matrix, like wounding, say one has a cut in one's hand or something, then the fibroblasts have to transform to specialized cells, which are called myofibroblasts, which have a phenotype somewhere in between the unactivated fibroblasts that are normally there under normal conditions and smooth muscle cells. And these cells are especially well adjusted to use their contractile forces to close the wound and to secrete matrix components so that they can heal damage to the tissue. So now, why is it important that the matrix be maintained in this steady state? And the reason is because the mechanical and biochemical properties of the matrix actually determine the form and the function of the cells that develop within the tissue. And a very good way of a very good demonstration of this fact is shown in the Engler paper that I have cited here. And what they did was to put stem cells into matrices of different biomechanical characteristics, soft matrices, stiff matrices, and so on. And what they found was that the mechanical properties of the matrix determined how the stem cells developed into mature cells, into the mature phenotype. And for example, they showed that cells that had been put into a matrix that was very soft, that mimicked the properties of nerve cells in brain, those cells developed into nerve-like cells, whereas cells that had been embedded into matrices that were very, very stiff, like bone, developed more into bone-like cells, and stem cells that were in matrices that were more like muscle developed into muscle cells. So that shows that the properties of the matrix determine what kinds of cells the stem cells develop into, and therefore, in order to preserve, say, the bone type or the muscle type phenotype, then the matrix itself must be maintained in the appropriate biomechanical state. And that maintenance is the job of the fibroblasts. As I pointed out before, fibroblasts under normal conditions can use small contractile forces and secrete matrix components such that small perturbations in the state of the extracellular matrix can be adjusted to get the matrix to have the optimized biomechanical properties for the function of the cells in that tissue, but that if there is a major perturbation, such as a wound, then, as I pointed out, the fibroblasts have to undergo a phenotypic transformation into myofibroblasts, which are, because of their enhanced contractility and ability to secrete matrix components, especially well adapted to healing wounds. And this is very well described in a relatively old, but nevertheless very informative review by Boris Hintz, cited here. So now, what I want to focus on today, though, is a model for how normal fibroblasts, under conditions in which it's necessary for them to adopt the phenotype of a myofibroblast, how that transformation might be going ahead, how it might proceed. And the first point I want to make, well, let me go back a step and say that the model that I have developed to try to explain this is called the cluster model, and you'll see why that is in just a minute. And the first point I want to make about this, though, is that myofibroblasts, because they are attached to the extracellular matrix and can contract the matrix using their actin-myosin stress fibers and cytoskeleton, that's one of the ways in which these fibroblasts can control the stiffness of the matrix. So that, consider a myofibroblast, which is attached to the matrix. It's going to exert a contractile force on the matrix. That contractile force is going to cause a slight stiffening in the matrix. And the first point I want to make about that is that, somewhat surprisingly, the stiffening effect of this contractile effect on the matrix by one or a few myofibroblasts in a particular location extends over a surprisingly large range, but nevertheless, the range is still limited. So the stiffening properties of one or a few myofibroblasts in a local region of the matrix can stiffen over distances approximating 100 microns or more. So the point about that is that if you have a few or one myofibroblast in a particular place, it'll stiffen the matrix to over a large but nevertheless limited range. Now, the second point is that the stiffening of the matrix, and this is a crucial point, the stiffening of the matrix actually increases the probability or increases the rate at which unactivated fibroblasts, normal fibroblasts within the domain, transform into myofibroblasts. So the more the matrix is stiffened by the contractile forces of the myofibroblasts in the area, the more nearby unactivated fibroblasts will be promoted, the probability of their transformation into myofibroblasts will be accelerated. Now, the kind of corollary to this assertion is that if you have a few myofibroblasts in a local region that are exerting contractile forces on that region and causing it to stiffen, then I am supposing, and I think this is a very natural assumption, that the more cells you have exerting that force in that local area, the greater the extent of stiffening, and therefore the higher the acceleration of the transformation of nearby normal fibroblasts, unactivated fibroblasts, into activated or myofibroblasts will occur. And this really leads to a positive feedback system, so that you have a few myofibroblasts in a local region exerting force, increasing the probability of, or maybe increasing the rate at which fibroblasts are transformed to myofibroblasts. Then you have more myofibroblasts exerting contractile force. That causes a still further acceleration of the rate at which nearby fibroblasts are converted into myofibroblasts. And so I think you can see then that the more fibroblasts are transformed to myofibroblasts, the more rapid is the conversion of other fibroblasts into myofibroblasts in that local region. So it's a positive feedback system. And I think you can also see that that means that there's going to be an increased number of myofibroblasts in the local area. So the myofibroblasts are going to form a cluster. That's where the name of the model comes from. And this cluster of myofibroblasts is going to grow in this local area due to the positive feedback. Now, a minor point, which I also allude to in this slide, is that the more myofibroblasts you have, as I pointed out, the more rapid the conversion of further nearby fibroblasts into myofibroblasts will be. But the very first cell, that very first fibroblast that gets transformed into a myofibroblast, that is undergoing this transformation without the accelerating effect of increased stiffness due to pre-existing myofibroblasts in the region. So in other words, the transformation of the first fibroblast in the cluster is going to be slower and less probable than the transformation of all the later fibroblasts into myofibroblasts. And so I make a distinction between the rate of the very first fibroblast, and I call that the nucleation of the cluster, and say that that's going to be slower and we'll have a smaller rate constant describing it. That's going to be slower than the acceleration, than the transformation of the later cells that join the cluster, and the rate at which that occurs will increase. The rate at which a fibroblast will be incorporated, will be transformed into a myofibroblast and incorporated into the cluster, that will increase as more and more fibroblasts get transformed into myofibroblasts, as more and more myofibroblasts exert contractile forces on the matrix. And this in turn accelerates the rate of transformation of nearby fibroblasts into myofibroblasts. Now, one thing that I'm not going to talk about, but I should at least mention because it is important, is that in addition to these mechanical enhancements by contractile force and increased stiffness on the transformation of fibroblasts to myofibroblasts, in addition to that, it is also important that various kinds of soluble cytokines elaborated by cells also accelerate and control the rate of transformation of fibroblasts to myofibroblasts. And TGF-beta-1, transforming growth factor beta, is a major cytokine that, among others, is involved in this process. So I mention that because it is important, and TGF-beta is important in the transformation process that I'm talking about, but I'm not going to talk about that. I'm not going to talk about that further in this talk. Okay, so the idea that I'm talking about here is shown in this kind of slide about the mechanism. Namely, that let's call N of J the number of clusters in the system that have J myofibroblasts in them. So let's suppose that you have a cluster with J myofibroblasts, and there could be several of these, and then you have some fibroblasts that are unactivated, just ordinary fibroblasts. And if this fibroblast is close to one of these clusters, then that will increase the rate, shown using a kind of chemical nomenclature, the rate at which this normal fibroblast gets transformed into a myofibroblast and gets incorporated into this cluster to form a cluster of J+1 myofibroblasts. So that's how clusters grow, by incorporation of our transformation of nearby fibroblasts into myofibroblasts, so that they are then included in the cluster. So in order to discuss this process, I've defined a kind of chemical, a kind of mechanism for the transformation, which is modeled after aggregation reactions that occur in chemical reaction systems. And as I pointed out before, the number of clusters in the system that have J myofibroblasts is called N of J, and the rate constant, you'll see what I mean by this in just a minute, by which a fibroblast is incorporated into a J-containing cluster, I call that K sub F of J. Now, K sub F of J necessarily must get bigger as J gets bigger, because as I pointed out, the bigger J, that is to say, the larger the number of myofibroblasts in the cluster, the greater the contractile force in the local region of the cluster, the greater the stiffening, therefore, the greater the stiffening of the matrix in the local region of the cluster, and therefore the greater the rate at which nearby fibroblasts are converted into myofibroblasts. So that's the formal way that I'm treating the model. And then just for completeness, I have to worry about the reversal reaction by which a myofibroblast in a J cluster is converted back into a normal fibroblast. And I call the rate constant, as you'll see in a moment, for that K sub B of J. Okay, so here are the individual reactions. And now think about this in terms of just a chemical reaction system, because that was the model that I used in developing this model. So what I call the propagation reaction, that is to say the growth of a cluster of myofibroblasts containing, say, J cells into a cluster containing J plus 1 cells, or in this case, J minus 1 to J cells, is the propagation reaction. It has these rate constants that I defined just a moment before. But there's this special nucleation process where the rates in transforming a fibroblast to a myofibroblast without there being any pre-existing cluster nearby has these special rate constants, sigma F and for the forward reaction, sigma B for the reverse reaction, and the magnitude of the sigma rate constants are much smaller because the nucleation process is much slower than the propagation reaction. I'm not going to dwell on this, but it's important to make that kind of a distinction. So again, here's something that I also don't want to spend a lot of time on, but just for completeness, I'll point out that the reaction mechanism for this, say here, you have a cluster that has 2 myofibroblasts in it, and then there's a rate constant to convert a nearby fibroblast into a myofibroblast, and that process is going to have the rate of that process is going to be the product of the special rate constant, which takes into account the fact that there are 2 myofibroblasts in the cluster, times the number of unactivated normal fibroblasts that are in the region, times the total number of clusters that have 2 fibroblasts. And all these reactions, there's a sequence of these reactions such that the cluster grows from 2 to 3 to 4 and so on. So that's the basic idea. And again, just for completeness, I don't expect you to read or understand all this, but basically this is just an example of the chemical, of what are analogous to chemical rate equations. In this case, they refer to cells in clusters. So these are the rate equations for the transformation of fibroblasts into myofibroblasts and the growth of clusters according to this model. Okay, now it's important then to realize that the properties of the transformation depend very centrally on how the rate constants are formulated, and I'll show you this in a moment. I have to formulate the forward rate constants in a way such that the rate constants increase with the size, the number of molecules in the cluster. So that's important. And the larger important point here, though, is that the distribution of the clusters over the course of time depends strongly on the nature, the parameters, which you'll see in just a moment, how those are selected, and the form of the rate constants. And I'm going to emphasize in this case a contrast between cooperative and non-cooperative mechanisms. And by cooperative, what I mean is that the cells that are transforming from fibroblasts to myofibroblasts are interacting strongly in such a way that they tend to undergo this transformation together. So the important point for the cooperative systems is that the cells are interacting and are encouraging one another over this process. And that's now contrasted to a non-cooperative model in which the cells transform independently. So I'm contrasting this with a case in which the cells don't interact with each other, but just transform independently. And I'll show you some examples of that. So now here are some examples. So it's crucial now that we define forms for these rate constants. And so here's an example of a non-cooperative system in which the forward rate constant and the reverse rate constant are just taken to be constants. And they don't depend at all on J, and therefore the number of cells in the cluster has no bearing on the rate at which the cells undergo the transformation from fibroblast to myofibroblast. And so you can see now that this would be a non-cooperative system. Now here are two examples of cooperative systems of rate constants for cooperative systems. So there's a rate constant, a general rate constant for that is the same constant for all the clusters. And I'm supposing here, and this is an extremely cooperative representation, that the rate constant increases as the number in the cluster times the stiffness parameter, S, which measures the strength with which each cell contracts the matrix around it. And that parameter, J times S divided by the total number of cells in the region of the matrix, is taken to the jth power. So you can see now that if J times S over XT is a positive number, and J just keeps getting bigger and bigger as the cluster size grows, then the rate constant, K sub F of J, is going to grow rapidly. And this leads to a highly cooperative case, because as you get to larger and larger cluster size, the velocity of transformation of fibroblast into myofibroblast gets bigger and bigger and bigger. And so the propensity of cells to transform into larger clusters becomes greater and greater. Now, another similar representation is exactly the same, except instead of having the variable J be the exponent, you just have a constant Q. And so this is also cooperative because the rate constants increase with J, but not as rapidly, because Q is a constant. It doesn't keep getting bigger and bigger with the cluster size. So now, having gone through this, I want to emphasize a general principle, which is that the more rapidly the rate constant, the forward rate constant, grows with J, the stronger the acceleration of the transformation and of fibroblasts into myofibroblast. And therefore, the more cooperative the transformation is. So this gets back, this is a kind of or conceptual illustration of the positive feedback idea that I talked about just a few minutes before. Namely, that as the clusters get bigger, the transformation probability of fibroblasts grows. And so you have this positive feedback system. Okay, now here's an example of the distribution of the different cluster sizes for a non-cooperative model. So what I did then was to select something I say, and therefore, as I pointed out to you before, the clusters don't interact with each other, and they just therefore grow independently. And so therefore, the probability of these different size clusters are not very different from each other. I could have chosen it in a way that they would all be exactly the same, but just for a better illustration, I've made it so that they're slightly different from one another, but they're all pretty close to each other. So this is an example of a non-cooperative case. And you can see that if you have an actual cell system that in a tissue that's undergoing this transformation, you can look at all the clusters, measure the sizes, the numbers of cells in the various clusters, assuming there are clusters. And if they're all pretty similar to each other, that's an indication of a non-cooperative process. And that's now contrasted to a system in which there's a very strong interaction, so among the different clusters. And here, this shows that. So down here, for clusters that have only clusters of only unactivated fibroblasts, and say clusters that have a single cell of myofibroblasts. That's a kind of simplest possible cluster. I've adjusted conditions so they're about at the same level, and I've chosen the parameter values such that it turns out that there's a high probability that you'll have larger clusters too. Now, because of this highly cooperative nature of the system that I've set this up to have, what you find is that there's a substantial probability under some circumstances where you have a substantial representation of either unactivated fibroblasts or very small clusters, and you have a substantial probability that you have a distribution of very large clusters, but then in between, almost nothing. So it's like an all or none. The cells either are present as very small clusters or present as very large clusters, but very few clusters, but the probability of clusters with an intermediate level of cells is much smaller. And so there again, if you had a cell system in which this mechanism was correct, then you would see a division between very small clusters and very large clusters. Yes.
Sorry, Elliot. Sorry. We just only have 5 minutes. Okay.
Okay. I only have about 5 minutes to talk about, because all I wanted to do was to show that the model could be set up in ways in which you could predict, in this very simple case, either all or none or uniform distributions. And the kinetics can show this as well. I won't dwell on that. But now I want to get to the main points of all this. And that is that the model makes experimentally verifiable predictions. And so what that means then is that in either natural tissues that are undergoing a myofibroblast contractility, so say tissues that are models for fibrotic tissues, or else in engineered tissues in which you have myofibroblasts doing this, then you can measure what the distribution of cluster sizes is, and from that even make estimates of things like this exponent that I was talking about and the rate constant. And so again, I want to emphasize the important thing is that the model makes predictions, and one can verify or disprove the presence of this mechanism for the production of myofibroblasts by looking experimentally to see whether these predictions are fulfilled. And there are various biomechanical questions. Of course, the main thing is, do you see clusters, either in engineered systems or in real systems? And how do the distributions of cluster sizes behave, as I described before? But then the biomechanical questions that are raised by this is, how can we regulate the stiffening? How can we reverse stiffening? And that's very important because fibrosis in many different organs, like the heart, the lungs, liver, cirrhosis of the liver, have very bad health consequences for very many people. So the idea then is to try to see whether this model can lead you to find ways of retarding biomechanical stiffening due to the transformation of fibroblasts into myofibroblasts. And as I emphasized before, experimental tests. Do you see clusters? What is the distribution of the cluster sizes, and so on and so forth? That will determine whether or not this cluster model is correct, does bear on reality, and if it does, will provide you with clues about how you might be able to regulate the formation of clusters and therefore the progress of fibrosis in tissues and scarring. And I think that's basically all I have to say about this. I'm going to stop at that point and I'll be available if anyone has any questions for the future. So, Vahid, that's the end of my talk.
Thank you so much, Professor Elliott. Thank you for joining us, have a lecture for us. On behalf of me and the chairman of the Congress, Professor Rezayat, and Dr. Ghazi, that executive manager of the Congress, I really appreciate you and your teams, Professor Janine, Dr. Farid, Dr. Shakiba, and everywhere that really impressed me and all of the participants, but interesting and new era of the fibroblast tissue and engineering of the about the drug and pharmacology. I want to talk about effect of pharmacology and drug screening, but we don't have any time. So I thanks from all the participants that participate our symposium, and let's start the next symposium. Thanks, Professor Elliott. Bye.
Thank you. I wish I were there in person, but I can't be. Sorry.